I will turn it over to Professor McGuire to introduce today's speaker.
Alrighty.
Oh my.
So for those of you who don't know, this is Skip's finishing talk. Right, so Skip gets the longer than usual introduction, although some of you heard me give the Barry Sonogo introduction last week, which was also long, but it was also a special event. So, but Skip's getting a fairly long introduction here. So it's definitely my pleasure to introduce our speaker, Phillip Skipwith, the MBZ's favorite curmudgeon. Yeah, everybody gets it. So Skip is a Jersey boy. I'm going to give you some background on Skip, some of which I just learned today. So you all get to learn some things about Skip, some of which I didn't know until literally like an hour ago. So I did know Skip was from Jersey. So Skip's a Jersey boy. I don't know if that has a connotation. But so he grew up in a town called Vineland in South Jersey. And when I asked Skip what town he grew up in, I thought he said Violent Land. And honestly, if you've ever approached Skip from behind and startled him and seen his reaction, it wouldn't surprise you if he actually came from a town called Violent Land. But in fact, it's actually Vineland. And Skip told me that Vineland is kind of this nothing town. Nobody knows about it. Nothing is going on there. And so for a kick, I thought I would just look up Vineland online and read about it. And I found a website that indicated like the latest news from Vineland. And the latest news from Vineland started, the lead story was, season 95 starts at Jim's Lunch, with a link to watch early birds get the burgers. So perhaps you're right about Vineland. So like me, Skip went to his local state university, and his local state university was the Richard Stockton College in South New Jersey, which is now called Stockton University. So it's a university that only formed, I guess, in like 1970 or so. So it's not a long-standing one, but there are a lot of people that are going to this university. In 2009, he enrolled in the master's program at Villanova University, where he worked with Aaron Bauer and Todd Jackman. And for those of you who don't know, Villanova has kind of become a main line from Philadelphia to Berkeley to our program. We've had a bunch of students come out of that program and join the PhD program here. So including Skip and Dan Portick, who many of you know, who finished recently. Also current students, Ben Karen and Jackie Childers, all of whom came through that program. And interestingly, I thought it was interesting that Skip didn't actually decide to go to Villanova based on any kind of incisive decision, that this is the best master's program in herpetology, and so I'm going to go there. It turns out that what Skip really wanted to do when he was applying for graduate programs was to do paleontology. And for those of you who know Skip, you know that he actually is like an encyclopedia of paleo, as well as an encyclopedia of herpetological knowledge and everything else too. It seems like he knows everything about vertebrates. He knows a lot about vertebrates generally. So he actually applied to some other programs to do paleo work. He didn't get into those programs, and Villanova was his stopgap. It was kind of his backup plan, and he'd been working on red knots, and so he applied to go there to maybe work on bird behavioral ecology. But before he arrived there, he actually got in touch with Aaron and pitched the idea, I guess, of doing a paleo herp project, which was approved. And then he showed up there, but when he got there, Aaron decided, no, you should work on molecular genetics of herps. So the rest is history. And I tell this story because I think it's a really interesting story of contingency and happenstance. It's a history that's a lot like mine. Not everybody who gets into this field ends up doing, like following a really directed course where they make all the right decisions in terms of which programs to go to. So for myself, I went to San Diego State University, which also had a great master's program in herpetology. But the only reason I was there was because I was a business student. I decided to pursue a business degree, and the other local university in San Diego that I could have gone to, UCSD, didn't have a business program. So I just happened to end up in this place that ended up setting me on the right course. And so me and Skip, we have that in common. I think that for I tell this story in part for undergrads, because I think that you never know if you're on the right path, but there are a lot. You just meet the right person at the right time, and suddenly it changes everything, and we can all wind up in the place that we want to be, in the future. So Skip and I share that, which I think is quite cool. So once Skip landed in the Bauer and Jackman labs, that really set him more on a direct course, a successful course in academia that he's on right now. He finished there, he came to Berkeley, joined my lab in 2011, and he essentially continued with the research that he was working on as a master's student there. He's developed and expanded it. He was super successful in our program. At the time that he graduated, he already had 7 publications, with more publications in the pipeline. And so things have gone really well for him. Once he just finished a few months ago, he actually got 2 postdoc opportunities. So he applied for the American Museum of Natural History Richard Gilder, I don't know if they label it that way, the fellowship. It's the Gerstner Fellowship. So he applied for this Gerstner Fellowship at AMNH, which is a difficult one to get, and he was the top choice among all their candidates. And he also applied for a National Science Foundation postdoctoral fellowship, and he ended up deciding to take the NSF postdoc, but he took it to the AMNH, where he's working now with Frank Burbrink. So he's, interestingly enough, he's back in Jersey. So he's living in Jersey now and commuting into the AMNH each day. I was going to say that he's back in violent land. So, the last thing I want to say is that, I tried to keep this pretty light and make a few jokes, but I know that it wasn't an easy course for Skip to go from Vineland, to a PhD at Berkeley and an NSF postdoctoral fellowship. I'm super proud of you, Skip, and I'm really happy that you're here to give this talk.
Thank you, Jim. It's been a pleasure to be here for the past 6 years. I want to thank everybody for coming out to see me talk about my life's work, I guess. And hopefully I won't disappoint you. So, yeah, can we get the lights turned off in the front, actually? That's possible. Good enough. All right, so I'm today we're talking about my dissertation work, which is, like Jim said, a continuation of my PhD. It's already going. So my interest started off with systematics. I've always been interested in trees, understanding how different species are related. But it wasn't until I came to Berkeley that I thought, like, I just don't have to make trees. I can actually do some pretty cool things with trees. And by trees, I mean phylogenies. And these trees are, for any field, are basically where we start. Any question we ask about what things are doing ecologically, you need a phylogeny to really test these things. The behavior, it doesn't matter. And for me, I'm really interested in the generation of biodiversity. So I'm just going to jump right into it. It doesn't take, a systematist, anybody who knows anything about animals, or animals or just organisms in general knows that diversity is not evenly distributed across the tree, a tree of life. And of all the millions of species out there, some are really diverse, some are not. And what I've always been interested in is first, what are the patterns of this diversity through deep time? And what are the processes that have generated this diversity? So there are a lot of factors that can factor into diversity, but here on this first slide, I just have birds. There are 10,000 species of extant birds, such as crown birds. But even in this slide, there are 10,000, each one of us can look at a bird and say, that's a bird. There are like, more, you have to fact check me on this. 3,000, what's 3,000 or 4,000 species of passerine, is that right? Something like that?
6.5.
Yeah, okay, of course. Parrots, there's 300 species of parrot. There's 20 or 30 species of pelicaniform bird. And then there are 20-something ratites, right? So these are like near the base of the bird tree. And then you would go immediately down the tree of extant tetrapods, and their sister taxon are crocodilians. And there's 23 species of extant crocodilian, right? And crocodiles used to be super diverse back in the day. You used to have running crocodiles, you have crocodiles that were in trees, or doing all sorts of crazy things. But now you only have 23, and most people looking at the slide will only know that there's crocodile, maybe alligator on there. I'll tell you right now, there's 2 species of crocodile on here from Australia. But most people wouldn't know that, right? So there are sister taxon birds are super diverse, crocodilians are not diverse. And there are lots of processes that can lead to the sorts of symmetry in the tree. So they can be intrinsic or extrinsic. They could be something to do with ecology. They could do with like something that's off the top of my head, like generation time, right? Or extrinsic properties such as climate change, vicariance events, extinction. In the case of crocodilians, right? So crocodilians experience a ton of extinction through the past, and not just one bout of extinction, multiple bouts of extinction through deep time. There are a number of traits in terms of phenotypic diversity. There's this long-standing hypothesis that diverse clades are going to be phenotypically diverse, and there's a number of reasons for thinking that. One is high punctuated equilibrium, where you have or character change is concentrated at speciation events, right? So it intuitively makes sense. Or that as if you're thinking of like the adaptive radiation, Schluter's definition of adaptive radiations, you find that, or you think that clades that are colonizing new areas, either speciation rate or phenotypic diversification are piggybacking off of one another. As you're diversifying along one axis, you're going to create diversity along other axes. And now we have the computational tools to really test this analytically, and that's a big part of my thesis. And part of like a sub category of this would be convergence, right? So I'm really interested in why you have different distantly related taxa that look very, very similar. Now, most people, like I work on lizards, they're not birds, they're not crazy, not flying around making noises, right? But they are incredibly diverse. There are as many species of lizards as there are birds, right? There are twice as many lizards as there are mammals in the world. But one of the things that I'm interested in is why, or at least the extent of replicated phenotypes across the tree of life. And convergence presumably is due to, or at least true convergence can only be due to similar selective regimes selected for similar traits, and that's due to ecological similarities. So just really quickly, among vertebrates, like some of the two best examples where you have replicated phenotypes would be Greater Antillean anoles, where on each of the islands you have, where they are more or less monophyletic, but then their most phenotypically similar taxa occur on nearby islands, right? So there are these multiple invasions of the same ecology from different distantly related groups that are replicated. You see this in South American cichlids and African Rift Lake cichlids, and I wanted to test this in my focal taxon, which I'll get to in a minute. But similar phenotypes can also be not due to selection pressures, but also due to shared ancestry, through plesiomorphy, or through like selection upon unrelated traits to expectation, right? And disentangling the two up until recently has actually been very difficult. You usually have to rely on a priori assumptions that these things fall into the same category. So I'm going to talk a little bit about my system first. So I work in Australasia. I've always worked for my entire graduate career. I've worked in Australia and the areas immediately surrounding Australia. So Australia, is a really interesting place because I always refer to it, I'm not the only person who does it, I'm stealing this from the literature. It's kind of a continental island, right? So it's been isolated since the late Cretaceous. It hasn't had any land bridges except in New Guinea for most of its geologic history. It's also a major origin of our center of biodiversity for a lot of groups. So mammals, marsupials, the bulk of mammal diversity in Australia are marsupials. They are endemic to Australia. Passerine birds are probably originated in the general vicinity of Australia and surrounding areas. But then you look at like Swami Reptiles, right, which are by far the most diverse vertebrates and terrestrial vertebrates in Australia. There's over 1,000 species of squamates that include snakes. It's a weird conglomerate, right? So it's not like they all got isolated there. They're monophyletic. And what I have here, there's over 50 families of squamates. I've pruned down to the 13 families that are present in Australia. Underneath the family name, I have the number, it's a ratio of the number of species present in Australia and then the total global diversity of that family. And you'll see on the right, you really can't read this, but on the right I basically describe where they came from and when they got their overall dispersal. For all the clades except for 3, they are recent invaders during the Cenozoic or very late Cretaceous. And that one exception is the coolest group of vertebrates in the world, diplodactyloid geckos. Geckos are the best lizards, by the way. These guys are the best geckos. I work on diplodactyloid geckos, and these guys are actually remnants from an ancient vicariance event that separate East and West Gondwana. Their immediate sister taxon are the geckonoid geckos that have subsequently invaded Australia from Asia. But diplodactyloids are the one exception where they have basically always been in the region, as far as we can tell. So what makes diplodactyloid geckos unique? So the sister of all other geckos are 1,700 species of gecko in the world alone. They encompass almost a quarter of lizard diversity. They separated sometime in the mid-Cretaceous, mid to early Cretaceous from other geckos. There are 3 families, the Carphodactylidae, but I throw these names around a lot, the Diplodactylidae and the Pygopodidae. And these are all encompassed in the Diplodactyloidae. And they're endemic to Australia, Asia region. And even though they only encompass 12% of gecko diversity, they encompass all of the morphological diversity in geckos. The geckos are crazy diverse. So you have little guys like this, like your little generic trash gecko, and then you have limbless geckos that, and limblessness only occurs once. So people look at these things, and some of them mimic elapid snakes in Australia, so they're super diverse. So this is just the region of distribution. You'll just notice by the numbers listed by the family name. Australia is a major epicenter of diversity, but there have been 3 independent island colonizations. There's been 1 colonization from the Pygopodids, which are the limbless forms, to New Guinea. Up until recently, New Guinea was connected to Australia. This is actually a low inland sea, the Carpentarian Sea in the north. And then there have been 2 colonizations by the Diplodactylidae to New Caledonia and New Zealand. And I'm just going to go a little bit into the geology and the relevance because we'll get a little bit into more of that. So New Caledonia, Australia is separated from Western Gondwana during the late Cretaceous, but this area is part of the Inter-Melanesian Arc. New Guinea and Fiji, Vanuatu are all part of the Melanesian Arc. So a lot of these are actually oceanic islands, or the coral atolls, basically, that have since expanded, volcanic islands. But New Caledonia and New Zealand, along with the Norfolk Islands and Lord Howe Island, are part of this sunken landmass called Tasmantis, or Zealandia. It's been in the news recently. And this was actually attached to the eastern margin of Australia up until about 80 million years ago, in which case it separated and then sank. And there's been subsequent orogenesis, mountain building since then. But up until recently, we thought that, that the New Caledonia and these islands are some of those biodiverse islands in the world, like you mentioned. They're basically second only to Madagascar and a couple other islands in terms of endemism, not biodiversity, endemism. It's like 90% of all plants are endemic to these islands. They're not found anywhere else. So for a long time, people thought that the biotas of these islands were actually due to an ancient Vicariance event. It turns out most of this was actually underwater until the late Oligocene, early Oligocene or late Eocene. And now we're showing that there's now evidence supporting that almost all of the biota of both islands are actually relatively recent overwater dispersalists. So here's just a simplified phylogeny of the Diplodactyls with each of the families colored in non-colorblind friendly colors. But yeah, so this is just to show the bulk of the diversities in the Diplodactylidae. The Pygopodids, I'm going to reiterate, are limbless. The Carphodactylids and Urine, they have limbs, but they don't have toe pads. So they don't have the signature toe pads of geckos. And then the Diplodactylids, the bulk of the diversity, all have toe pads. And what I want to get these, when the pictures are not to scale, but as I want to say, not all geckos look the same, right? So they're incredibly diverse in shape and in coloration, and subsequently in ecology, and also in body size. So take a guess at who that is. So you can see that some of the smallest geckos in the world are in the genus Crenodactylus. There are some way smaller geckos actually in the Dominican, but these guys are tiny. They're the size of a quarter. And also the largest extant gecko, also Wallace's clades. So these guys are the length of my forearm. And then the longest geckos are the limbless geckos in New Guinea, Lialis, and then some burrowing forms also small. So there's actually quite a bit of variation of body size across the whole group. There's also quite a bit of variation in terms of osteology. Your generic gecko, really lightly built skull with small peg-like teeth. But then, in some of the island forms, you have these guys that have really robust teeth. And these guys are the top predators on the island, with the exception of introduced rats. These guys eat almost exclusively nothing but other lizards. Huge caniform teeth. And then you have some of the pygopods, which take cranial specialization to the extreme. So it looks like a big bird skull, where the skull actually has this frontal parietal suture that allows the skull to flex like a thing. And you can push on the snout, and the snout will bend, right? So and these teeth are hinged, so they can fold because they're eating other lizards. And it's a specialization you don't see in any other group of geckos. So my research interest was, one, building a new phylogeny, because phylogenies are cool. And there are phylogenies, there have been numerous attempts to address the phylogeny of this group because it's so diverse. But they've been either incongruous with one another or they have been really poorly supported. The molecular data is actually really not consistent across the groups. And I'm not going to go into the nuances of the phylogeny because it's boring. But to address some of my other questions, I needed a really robust divergence time estimate. So I built a new divergence time estimate and tested some of the biogeographic history of the group. But then I was really interested in was the tempo. First off, what are the patterns of diversity? And what is the tempo of diversification across the group? And I initially hypothesized that the two island radiations, major island radiations, New Caledonia and New Zealand, since they were probably overwater dispersalists, experienced explosive diversification in terms of speciation rate, and also explosive diversification in terms of rates of phenotypic evolution. So I wanted to test this in a rigorous way. No one's really tested this across the group. Also, there was a recent paper that came out, I panicked when this paper came out. It came out like in March, I think, looking at the phylogenetics of this group, and this paper made a strong argument that there was a mass extinction event. I'd say strong. They made an argument, it wasn't strong. Mass extinction at the base of the Diplodactyloids coinciding with the Eocene-Oligocene boundary. And the importance of this is because during this time, Australia was still moving northeast. And this also initiated what, people think of Australia as a big desert. At this time, Australia was actually tropical savanna, which is now restricted to northern Australia. But this initial movement northeast resulted in this changing of currents, and it was this cooling of the entire Australasian region. And a lot of groups, including birds and mammals, experience at least some extent of mass extinction. So they tested this for the diplodactyloids, and they actually found a signature of mass extinction at the base of the radiation and the early Cenozoic. And I also wanted to test, there are a lot of replicated phenotypes in this group, at least what I thought were replicated phenotypes, particularly among the islands, right? So you have these different forms that are superficially very similar. Some of these groups morphologically cluster together in phylogenetic analyses. And I wanted to test the incidence of convergent evolution using non-a priori groupings. So to do this, I tried to get the best sampling I possibly could. This is an example of the importance of natural history collections. And of the 213 described species, I was able to sample roughly 75% of species, and I sampled all but one genus. And previous studies have used small nuclear data sets or exclusively mitochondrial data sets in terms of molecular data. And I used ultra-conserved elements, which are, as far as a grad student, are relatively cheap. And you can get 5,000 loci. They're really designed for birds, but they have broad phylogenetic applications across tetrapods. So you can use them for mammals, other squamates, and amphibians. All lab work was done in-house, and I'm not going to go into any of the bioinformatics or the details of some of the downstream analyses, but I would see KB sitting in the audience. This would have been possible without KB. For most of us who are not bioinformaticians, yeah, people like KB make this possible. It's amazing, actually, the amount of work I was able to get done with his help. Lydia Smith, who hopefully is also here.
I'm here.
Thank you to Lydia and Kay. They help for literally both ends of this study. Always harassing them, asking them to do stuff, and I'd be like, Katie, I can't get this to work. And he'd be like, all right, I already did it already. It's in this directory, so it's great. And down the lines, the scripts were in Perl by KB or Dan Portick, who overnight became a programming whiz. So it's not in the books for me. So in addition to the molecular data, I actually worked extensively with physical collections, and I visited 10 collections in 3 different countries to get external ecomorphological data that test some of my diversification analyses and to test for phenotypic convergence. So I was able to sample about 148 species from the tree in these collections, 1,100 specimens, and I looked at traits related to dimensions of the head, the limbs, the toe pads, all things that are directly interfacing with their environment, including body size. And all of these were corrected for with body size by using phylogenetic least-squares regression, and depending on the analysis, either using the residuals from this regression or reducing dimensionalities to ordination with phylogenetic PCA. So I'm just going to jump into my results. Very briefly, it was like a really resounding success. When I initially started doing the phylogenetic analysis, or the lab work, I was like, this is going to be a catastrophic failure. A waste of NSF money. It worked way better than I thought it would. So roughly 50% of the reads that, or base pairs that pass filtering actually back to the UCE probes. I was originally aiming for 40x coverage. I would have been happy with 3x coverage, honestly. My average coverage was actually 220x. Which is incredible. I was actually shocked by this, with the exception of one sample which had 12x coverage. That's too bad. I'm still going to use it anyway. And of the 5,000 loci that I was targeting, we were actually able to get over 4,200 loci. So to my knowledge, this is the largest matrix in terms of species and loci for any group that I know of, in terms of the combination of the two. People have bigger trees in terms of species, but not with thousands of loci. And there's actually a fair amount of variation in locus length. Largest ones are about 1,400 base pair, probably due for trimming off those small loci because they're not really doing anything. And this is just a plot of coverage showing, organized by clade, showing coverage. There's actually quite a bit of variation in coverage, but all the coverage is very good. I just wanted to know that how informative ultra-conserved elements are. The name's kind of a misnomer. You think that they're just not informative at all. There actually is a strong relationship between the length of the locus and base pairs and the number of parsimony-informative sites. The longer the locus, the more parsimony-informative sites you have. It's not a strong correlation. They're more conservative than, say, exons or anchored tags or some of the new broad phylogenetic data sets, but there is a strong relationship, which is good, right? So for phylogenetic analyses, I did this multifaceted approach, both coalescent and concatenated analyses. I'm not going to go into the details. I did multiple concatenated analyses. They were unpartitioned just for computational. Can't really have a partition analysis with 4,200 partitions. The computer would die. Yeah, so I'm just going to show the results. The topologies are identical across the phylogeny. And I was initially, I heard spoken as when I initially pitched this project, I had multiple people tell me, you're wasting your time. This is stupid. I don't know why. Like, and it's not exaggeration. Actually, I heard from multiple people that phylogenomics are a waste of time at higher levels, right? Because we already have our mitochondrial tree. The tree's not going to change that much, even if it's poorly supported. You're just going to converge more on get better support for the same exact tree that we've already got. And I was initially worried because we actually have the familiar level relationships are actually almost identical to previous studies. So we have pygopodids rendering the limbed forms paraphyletic, with the diplodactylids being sister. You can't read this, and it doesn't really matter. But the take-home point is that we have the two island radiations, New Caledonia and then New Zealand, are not related to each other. Even though they are both occur on geologic islands that are geologically related to one another, they are non-monophyletic, which is not surprising. We actually have known this from poorly supported mitochondrial trees for some time now. Previous work from my master's thesis also supports this. But it was nice that the UCE tree was not giving me something wildly different. So in addition to the concatenated analysis, I also did multi-species coalescent, and most true multi-species coalescent is not really possible for data sets of this size. So we use a summary multi-species coalescent, which is in general agreement with typical methods like BEAST or Star Beast or SNAP or something. So to test the stability of the trees of my data set, just to estimate the number of loci it takes to get to the full species tree topology, I randomly split my lotus set in my data set. We'll try to explain this. I always get tongue-tied when I try to explain this. Into different size bins. So I had 10 bins. Right? If I get confused, somebody please stop. Or if you get confused, stop me. And I randomly sampled it and potentially resampled those bins. So I had 10 bins, 10, 20, 50, 100, 250, all the way up to 4,000 loci. It's sampled from my full data set 20 times that number of loci. Does that make sense? And then built a species tree for each one of those loci, and then computed the Robinson-fold distance between those subsample species trees to my full species tree to see how many gene trees it takes to get to my full species tree, because you'd expect that with the data set of the size, incomplete median sorting would be a problem. And it's not kind of nice because at about 500 loci, you recover 90% of the nodes in my full 4,200 loci data set. And after about 2,000, you stabilize at about 95%. So you get rapid convergence. It's not the same. I think with Dan's Afrobatrachian frog data set, it only takes 200 loci or something like that. He only had 1,000. So on average, they're more informative, right? But yeah, so the species tree is actually quite stable with a low number of low number of loci. But then I also wanted to do downstream analyses. I also wanted to get a robust phylogeny or divergence time estimate. Not necessarily compare the divergence times to other studies, but to actually do a different analysis. So to do this, most methods you can't actually do this with a data set of this size. So I did use a subset of my data set in a program called Mcmc Tree and PAML. It's a nightmare, but it works really, really well. And we did this on the astral topology. And the divergence times are mostly congruent with previous studies, but they're typically much older. And I think this is actually an artifact of how the models are working. I'll go into that a little bit later. But basically the point is the crown of this group actually goes back to the late Cretaceous. Previous studies have said it dates back to the Paleocene, but this goes back to the Santonian or something. And then the most important, interesting thing I think from the data tree is that this recapitulates the argument that the two island radiations, New Caledonia is up here and New Zealand's down here, actually vastly post-date the emergence of the islands. So this requires the argument that these guys dispersed to their respective islands independently during the Neogene. So for the people out there that were telling me that this is a waste of money, I wanted to see, A, is the tree better supported than previous studies? And is it converging on the exact same topology or very similar topology to previous studies? So to do this, I pruned the literature for other studies that had at least 100 species of diplodactyloid, and then computed Robinson-fold distances for their trees and my trees. So some of these are Sanger-based studies, and others are super trees where they have 90% missing data. So this is just the percent well-supported nodes, and there are issues with comparing across statistical assessments of node support, but I did it anyway. So my trees on average much better supported than the previous studies. So these first two bars, this is my concatenated tree. As you would expect, it receives higher proportion of support nodes than my coalescent tree, but both trees get better support than these 4 published studies. Right, so some of these are like only 60% of nodes are well supported. So this does improve statistical support across the whole tree. And that alone, to me, is not really the clincher. But I wanted to compute how similar these trees are to previous studies. So on the left, I have a bar for a direct comparison of my concatenated and my coalescent trees. So the two agree on about 85% of nodes, which suggests that there's a fair amount of incomplete name sorting. But then you compare both, there's the species tree on the left and the likelihood in the concatenated tree on the right to previous studies. There's actually a incredible amount of variation in this discordance. So they share anywhere between 75% of nodes to as low as 50% of nodes, basically. So some of these trees, they disagree on almost half the nodes. So I think that's actually a strong argument that phylogenetics actually do matter, right? So you can't just assume that you have your species tree. I think most people would probably agree with this. All right, so in addition to estimating the phylogeny, I wanted to do some actually really interesting things with estimating the tempo of diversification across the whole clade, and to see if there is rate heterogeneity. So to do this, I used a number of different methods. I'm not going to go into the details of the methods. All the methods are some derivation of a birth-death model. So the few exceptions are DDD, which tries to estimate diversity dependence, as there's a hard cap on the number of species you can have in a clade. TESS is complementary to the previous study I mentioned before, where they try to estimate mass extinction events, the relative importance of the mass extinction events in the clade. So I use TESS to see if, with this new timing estimate, if there is a mass extinction event, and when is it, and what is the magnitude of that mass extinction event. And then BAM was used to estimate rate heterogeneity, so clade-specific shifts and net diversification. In addition to that, I tried to see the relationship between net diversification and rates of phenotypic evolution to see if they are correlated like I hypothesized, and also the relationship between body-size evolution and the colonization of novel bioregions. So this is just a first pass. This by itself doesn't really mean much of anything. I'll just start with most clades show some signature of decline. There are a lot of patterns or processes that can lead to that. It could be decreasing speciation, it could be increasing extinction, it could be episodic extinction. And basically molecular data can't really tell you that. But almost, it's probably, for me, I should probably start with the most problematic thing. But almost all of these methods recover extinction rates that are near zero, which is basically impossible. But almost all of them also show signatures of decline, which, like I said before, is not unique. So just to illustrate this, I've simulated a pure birth tree, a Yule tree, and estimated the gamma statistic for this. And the gamma statistic just tells you a deviation from a pure birth process. So it's not significantly different from a pure birth process, which is what you expect. It's simulated to do that. So from my empirical data set, there's actually a very strong deviation from a pure birth process, and there's a strong signature of decline across the whole clade. So with BAM, unlike what I hypothesized that the two island clades, there's New Zealand and New Caledonia, I expected a rate shift in terms of speciation rate for these two clades. We actually find no rate shift, clade-specific rate shifts across diploidactyloids. Instead, there's actually homogeneity across, which I found actually quite surprising. So the background rate is actually quite high for all diploidactyloids, and BAM finds this actually this plot is misleading. It shows that there's some magnitude of increase towards the present, but actually that magnitude is actually quite small. This is just one tree from the posterior, so I wouldn't go by the colors for this one tree. But that speciation rate is actually quite constant. So this is a better display of this information. So up in the upper left-hand corner, I have plotted net diversification. These gray bars represent the 95% credibility intervals from the posterior. This is a Bayesian analysis, so I have set up samples from the posterior estimating extinction and speciation. And basically for each of the entire clade, each of these subclades, even though they're different ages, speciation rate is actually very constant across the clade. They don't deviate from each other, and it's very flat. Some samples, they imply an increase or a decrease, but Bayes factors, model comparison, reject any sort of heterogeneity. R Panda, this is a spectral density graph using maximum likelihood to estimate the number of modalities across the whole tree. So modality is analogous to a rate shift. And this is the single color, this is red, just shows that there are no rate shifts across the whole phylogeny, which is surprising to me. So the previous slide that showed mass extinction in the EEOC bound, or EOB, the Eocene Oligocene boundary, which would be right around here. I should have mapped it on here. I ran a complementary analysis with tests, and I ran 2 sets of model testing analyses. So this is a reversible jump Mcmc analysis where I resampled the data set. And basically we find very little support for a mass extinction event about 75 million years ago. It's like smack dab right in the Campanian. So it's the second to last epoch before the KT boundary. So it's the Campanian and then the Maastrichtian and the Cretaceous. So that predates the EOB by about 25 million years. So this suggests that the Eocene Oligocene cooling event had nothing, if there was a mass extinction event, for which I find very little support here, had nothing to do with that, and I actually have no idea what that means. And then with some more complicated stepping stone simulations, we actually find much stronger support for the exact same timing. And I'll go into that later on. So the take-home point is, though, is that this contradicts previous studies on when, if there was a mass extinction event, when it was. So how are traits evolving, and where are the relationships between trait evolution and net diversification? So to do this, I also use BAM and I also use OWI to test Hansen models of stabilizing selection for body size. And for BAM, I use 6 different traits on 3 different trees. So either the full tree, a subset of all 3 families, or a subset of just Carvidites and Diplodex for traits related to limbs. Pycopods don't have limbs, so I kicked them out for all comparative analyses that require limbs. And this is just a plot showing, because I showed you that figure with my awesome cutout, showing the range of body sizes, but I just wanted to show that really large body size is concentrated in 3 different clades. The island guys, the New Caledonian guys, and then the Pygopodids, which can be huge. And these bars are proportional to body size, if that's not obvious. So here's a BAM trait analysis on body size, and we find 2 rate shifts where they're like 2 orders of magnitude faster than the background rate. And that's once within the New Zealand clade, and once within the New Caledonian clade, where these guys are either very large, or they're related to very small guys in the case of the New Caledonian guys, or they're just very large in the New Zealand guys. So we have 2 rate shifts and body size. And out of the 6 traits, only 2 other traits actually show heterogeneity. All the other traits show homogeneity in the rate of diversification. But it's not shown in the same clades, which is also surprising to me. So head size shows a large increase in the rate of diversification in the Pygopodids, the limbless guys. This tree on the left shows head size, and the Pygopods are here, right around the base. They show an increase in the rate of head shape evolution, relative head size. This is corrected for body size. Relative head size. And these guys are limbless, so it's probably related to loss of limbs as you get longer. And then with toe pad width, so this is corrected for body size as well, the 4th toe pad, and these Australian guys, these guys are all terrestrial. This clade includes Leucasium, this is one of them. These guys, if you can't really see it, they actually have really tiny toe pads, have increased rates of toe pad shape evolution. And to also determine the extent, like you could have different shifts or different clades shifting along the same axis, but are they doing it in the same way? So to visualize this, this is a BAM macroevolutionary cohort analysis, and this looks like a confusogram, probably. But basically, this is the full 180 species data set. Here is the same tree. These are both the same tree, and you can see the 2 clades that have rate shifts are colored differently than the background rate. Each one of these cells corresponds with a pairwise comparison between the 2 trees. The trees are topologically identical, but warmer colors indicate from the posterior samples that they share the same rate regime. And then cooler colors imply that they are not in the same rate regime. Does that make sense? All right, so basically it shows that these 2 clades, not only are they not in the same rate regime as other clades that are not experiencing rate shifts, they're not in the same rate regime for body size with each other, which implies that they're diversifying on body size to different magnitudes. And that makes sense because the New Caledonian guys, the New Zealand guys have a much higher rate of body size evolution estimated by BAM than even the New Caledonian guys. Even the New Caledonian guys are much bigger, which is actually quite cool. And I'm not going to show it because it's boring. But for the other 2 traits, you see a similar pattern, where different clades are under different rate regimes for head size evolution and toped width evolution. So using Hansen models of testing models of trait evolution in OWI, I wanted to see, based on 4 broad geographic categories, just in which region body size is evolving the fastest. And it's the 2, as you can see, it's the 2 island clades. So New Caledonia and New Zealand have the highest rates of Brownian motion body size evolution. So this is a multi-peak or a multi-rate Brownian motion model. There's no stabilizing selection here. It's just that each group is allowed to have varying rates of stochastic body size evolution. And New Caledonia clade and New Zealand are much higher. That's the take-home point, which I think is a nice confirmation of what you find with BAM. So now I'm going to jump to like a little subset of this data set, testing for actual phenotypic convergence. And this is actually kind of complicated, a little messy. So to do this, I use the same morphological data set, but instead of using just the residuals, I use the phylogenetic PCA scores. And like I mentioned before, I had originally hypothesized that the New Caledonia and New Zealand guys were convergent with each other, and that various ecomorphs between Australia, New Caledonia, and New Zealand had been converged upon more than once. And I had 6 putative a priori ecomorphs, which I thought represented distinct categories, right? They're doing the same thing, but they're in different clades. And then 8 broad ecological categories that I'll explain in a bit. I reconstructed ancestral ecology using these 8 categories, using stochastic mapping and maximum likelihood, and then tested, just like I did for the previous section on biogeography, I tested rates of body size evolution for the 8 ecological categories. And then with ignoring the a priori groupings I had made, I used 2 different models that test whether or not there's the presence of convergence, and whether or not these fit in the categories I'd already described. I used surface and L1 OU. There's actually a whole suite of literature now saying that these methods are crap, basically. They don't work. They have a really, especially surface has like almost like a 90% type 1 error rate. And that's mostly an artifact of using AIC. So I don't know what to do with that. So here are the 6 a priori groupings I described. Up in the upper left-hand corner, we have generalized arboreal. These guys like live in trees and bushes. They have broad toe pads, small heads. We have this Rapiculus form. The Rapiculi just means living on rocks. They have really long legs, broad toe pads, and these broad, large, flat heads, so they squeeze into crevices. We have your generic little gecko, your scansorial ecomorphs, so they're just like a jack of all trades, living on multiple substrates, with broad toe pads and short limbs. And then you see these like super specialized terrestrial forms that are eating things like scorpions and spiders. They have these huge heads. Geckos are badass, by the way. so they eat anything that can fit in their mouths. And this is this original tail, so it's not a regenerated tail. So this is a really specialized ecomorph. And then you have another terrestrial ecomorph that has tiny toe pads and these tiny heads, and these guys like live in spider burrows eating termites. This only occurs in Australia. And then we have this limbless grass-swimming ecomorph, which is like a very broad category. So for the 8 ecological groups, we have arboreal, shrub-inhabiting, fossorial, grass-dwelling, ramniculus, grass-swimming, which are the limbless pygopods, Rapiculus living on rocks, scansorial, crawling around on anything, and then terrestrial. And what's really interesting is that it reconstructs both methods are identical. You get similar estimates for the posterior probabilities in the marginal ancestral likelihood states for both maximum likelihood and stochastic mapping. I'm just showing stochastic mapping. I'm talking really fast. And then so it gets relatively strong support for the ancestral diplodactyloid being living on rocks, Rapiculus, and there's been multiple transitions from Rapiculi to all these other ecomorphs, but very few transitions from other ecomorphs to Rapiculi, or to other not ecomorphs, ecologies, with the exception of scansoriality to Rapiculi, because a lot of scansorial things are also living on rocks, which I found very interesting. And then when you test these, the same 8 ecological categories, you find that arboreal things living on trees have the highest rates of body size evolution. And it's not surprising because some of these guys are like this big, some of them are this big. Right? So there's a huge amount of variation that's widely distributed across the tree. And then you have fossorial things, which are only found, truly fossorial things are only found in pygopods, and they're all small. So that it makes sense that they have the lowest rates of body size evolution. All right, so I'm going to jump into to my tests of convergence without a priori grouping. So I get actually 2 radically different answers. But this is a surface, right? This is what maximum likelihood. And what we find is that this is a super diverse group, as I've hopefully impressed upon you. But they find 28 distinct shifts in morphospace. So 28 distinct shifts in morphospace and 22 distinct morphological regimes. So there's 22 distinct spaces of morphospace, just if that makes sense. 6 of which have been converged about more than once. We have 6 regimes that are occupied by distantly related taxa. And then I also simulated trait data sets on the same tree for under a single state Brownian motion model, right? So that'd be non-convergence. A simple OU model with low rates of stabilizing selection, and a truly non-convergent model with no stabilizing selection. And then compared the proportion of samples in which they exceed what I see in my empirical data set. And basically what we find is that we don't have exceptional convergence at all if you have any level of stabilizing selection or just random trait evolution. But it's more than you would expect if there was no selection at all. Does that make sense? So, and the other thing that's really interesting is that you'll notice that all right, I actually didn't explain how to interpret this. All right. So the gray branches, the gray and black scale branches, those represent regimes that are only shifted to once, so truly non-convergent regimes. While any branch that's not gray or black that has a color are convergent regimes. If that makes sense. So almost a third of all the convergent regimes occur in the New Zealand guys and the New Caledonian guys. So basically the other groups don't really demonstrate much convergence. So it's the island guys that seem to be doing the most in terms of convergence. So I'll just briefly walk through some of the regimes that are detected by surface. You see some cute gecko pictures, I guess. So the first regime is this really specialized group I mentioned before that lives in spiral burrows, where you have these guys already have really small heads, and then you see 2 groups that actually independently evolve even smaller heads. These guys live inside termite burrows and are eating larvae. They have no toe pads, really fat, chunky bodies, and short little legs. We also see a regime of generalized convergent, generalized arboreal guys that occur in New Zealand and New Caledonia. Some of these guys, you see them and they look almost identical on the 2 island masses. They've got broad toe pads, intermediate sized heads. You see like a miniaturized version of this only in New Caledonia, where these guys have broad toe pads, small heads, and for a long time they were thought to be in the same genus. Turns out they're not sister taxa. And then we have this clade, or this ecomorph or form that appears both in Australia and in New Caledonia, where they're the true giants. They're really, really big guys. And these guys have all really broad toe pads. Hopefully you can see this on the Brachydactylus down here. Relatively large heads and these large, robust bodies with long legs. And then like an intermediate form, also found in New Caledonia and New Zealand, that are gigantic, have broad toe pads, long legs, and then their heads are almost a third. They're the exact opposite of like that first group I showed, where they have heads that are gigantic, they're almost a third their body length. And then lastly, this generalized terrestrial form that only occurs in 2 different lineages of Carphodactyla that have no toe pads. So it makes sense that they don't overlap in morphospace with any other group. L1OU, which was designed as a complementary analysis that doesn't rely on AIC. Instead, it uses a phylogenetic lasso to penalize rate shifts or state shifts along the tree. Gets sort of nonsensical results. Basically, it only recovers about half the shifts found by surface. And instead of 6 regimes, there's only 3 regimes. And they're all concentrated in New Caledonia and in New Zealand. These colors actually don't make any sense. This is actually an artifact of the program. This is actually a pathological issue with the analysis. But if you actually look at which clades shift upon the same optima theta, it's only between the New Zealand clade and the New Caledonian clade, which recapitulates that most of this adaptive evolution is occurring, at least the traits that I examined, is occurring within the island taxa and not the mainland taxa. So this is a lot of information. But I just sort of flew through. But hopefully I've impressed upon you the importance of phylogenomic data, or at least reassessing the phylogenetics of the group for which you've already been examined. The divergence dates that we give this clade I actually think are an artifact of me using Mcmc tree and most studies using Beast. And I think this is not an artifact of UCEs being slower evolving than other data sets. They're no slower evolving than most exons, right? But the way the 2 programs actually handle, most people who are using Sanger data sets are using Beast because they can handle small data sets. But it's simultaneously estimating the topology and the divergence times. MCMC tree doesn't do that. MCMC tree only estimates divergence times on a fixed topology. And the way Beast handles the multiplication of the true prior and the time prior can allow it to violate your calibrations, right? So you can actually get dates that are really, really young. So which I think calls into question a lot of, I'm not saying what other people have done is wrong necessarily, but I think people should maybe do some side-by-side comparisons between MCMC tree and beasts, because that's a little worrisome, because I've used both now, and I'm getting rather different divergence time estimates using the same calibrations and the same type of priors. This recapitulates the importance of both vicariance events and dispersal in this study, which is good. All right, so the two island clades are actually quite young. You can only have reached the islands by overall dispersal, long distance overall dispersal, sometime during the Cenozoic. And then the diversification genetics are actually really, really complex. So instead of having speciation rates and net diversification rates and trait evolution, rates of trait evolution that are correlated, which is what I originally hypothesized, and you would expect that, right? That something would get to an island and they would explode in terms of species diversity. That would be above the background rate. And they would also have a complementary increase in the rate of trait evolution. We actually don't find that, so the two are actually decoupled. And the fact that different traits are evolving at different rates for different clades implies that there's some level of both modularity and also constraint for certain traits for different clades. So just for an example, in the New Caledonian guys, you have the major axis of which there is an increase of the rate of diversification is body size. But the other traits are not having an increase in diversification in these guys, and I think that might have something to do with constraint. So these guys have like a little founder effect where they're getting to this island, they're diversifying only on one axis. You don't see them going to rocks. Their ancestors were probably also living on trees, but they're not living on rocks, they're not fossorial. So the one axis they can then stratify. You see this with the Anolis, where you have things that live high in the canopy and things that live low in the canopy within Anoles. You see very similar things with these island geckos. And the traits that are being that show rate shifts and rate of diversification and also show high signatures of stabilizing selection are also traits that are directly tied to diet and locomotion, right? So you have a small head, you eat small prey. You have a big head, you can eat whatever you please. If you have big toe pads, you're probably a better climber, maybe. And if you have long limbs, you might be crawling around on rocks or doing something weird. So this makes sense. This is not surprising. But this is the first multivariate data set to really be used for geckos in a comparative context, as far as I know. And convergence, while present, is not really rampant. I thought this would be like, this is better than the Anolis, right? So these guys are more diverse in terms of ecology, and this will be like a new model system for studying convergence in squamates. And I don't think that's actually true, but I think it's because their adaptive radiation is doing something very different than Anolis or something like cichlids are doing, where contingency is really important. These guys are invading different habitats, and each clade is doing its own thing. Right? Otherwise, you wouldn't have the diversity that you see. You wouldn't have limbless ones. There are no limbless Anolis. And I think that the Diplodactyls represent a conglomerate of both adaptive and classic non-adaptive radiations. So on the left here, I have Adura. These are about 4 different species of Adura. This clade is Australian. It's about 20 million years old. And other than pattern, hopefully everybody can agree that these guys look very, very similar. They have very similar proportions. The disparity is very low. They're all pretty similar in body size. And then we have the New Caledonian Diplodactylids, which are roughly the same age, about 20 million years old. And they're extremely diverse in ecology. These guys are all insectivorous, occasionally taking vertebrate prey. These guys, these giant guys eat nectar, they eat flower petals and stuff. Some of these guys are insectivores, living in like little burrows and trees. Some of these guys are like sitting on trees, pouncing on other geckos. They're like lizard panthers, basically. So it's just like really, really diverse in this, in terms of ecology. But you have this weird heterogeneity in terms of the ability to explore novel ecologies and morphospace that is not homogeneous across the whole tree. And I think that a lot of it probably shows pattern where it's not just one big adaptive radiation. You're not going to see extremely high rate shifts across the whole tree, but instead it's going to be like little pockets. So looking at it in a more holistic approach is actually really important. So that's it, basically. Obviously, this study wouldn't have been possible without help from my collaborators in Australia and my committee. You guys are awesome. And the labs, like a grad student is nothing without the community that they're in, both in terms of grad students and resources that are available. And I, 6 years, I was here over half a decade. And it's, it's such a long time. But it's, it's the McGuire and Bauer labs, the IB department, you guys are amazing. Grad students just made this experience, doable. Otherwise, I don't think I'd be standing here right now if it hadn't been for the support network that I have from both my superiors and my peers. my grad students. Museum collections, you know, none of this would have been possible if I couldn't, if people hadn't collected them, including my own collections and the work other people that I do, and the collections managers at different collections have made this possible. And like I said, I visited over 10 collections, and I'm not a programmer, I'm not a bioinformatician, I'm not a theoretician. I chug along the best I can and beg for help whenever I can, which is all the time. So you know, this wouldn't have been possible without software help from these people. And again, it's like the fake everybody, you know, no man is an island. And without taking any questions.
You need to file out first and then go ahead with the questions. Just look at the back in here.
So geckos are the best lizards. They've somehow managed overwater dispersal. Like, do you have any idea of like how that might have happened, or what is, like, you talked a lot about what's unique about this group, what's special about the ancestors that might have made this possible?
That's like a whole like rat's nest of problems. So I mean, for a long time, people thought, one of the reasons people thought that this clade was due to vicarious, I didn't really focus on that too much. But basically, up until my own work, like from 2011, people thought that this clade was actually quite old, right? So there were no divergence times estimates on the island radiations. It's because most geckos have calcareous eggs, and these guys have actually like pretty thinly shell they lack the calcareous eggs. So you see lots of overall dispersal in other geckos, but not in these guys. So one of the arguments is because they lack it, they couldn't do it. And frankly, I have no idea what traits. I don't think you need any special traits. I think you just need one gravid female, you know, to make it to these islands. And geckos aren't unique in this. Almost all lizard groups have some example of overall dispersal. I actually can't think of a single trait about geckos that makes them special. It's not like they produce more eggs, you know. They're not. Yeah. So the answer is I don't know. So. Jim.
How come to get the next plates on the beginning except for the one miles?
I think that actually has something to do with competitive exclusion from geckonids. So the group of geckos that you work on are much tougher than the group of geckos that I work on. So again, the geckonids aren't really diverse in Australasia in general. So once you get like east of Wallace's line, their diversity drops off a lot. But there are 3 genera that make it to New Guinea. So there's Gahyra, gecko, and Cerdodactylus, and they're all really diverse there in terms of ecology, not necessarily species. And I think that the Diplodex are just late in the game. And then the connectivity between New Guinea and Australia has been really intermittent. That sea has been there, or there's been a lake there sometimes. So I just think they just haven't been able to establish themselves. And that's also one of the reasons why the geckonids, when they did make it to Australia, they're not particularly diverse. So, yeah.
So you showed different clades have different amounts of morphological disparity and different rates of morphological evolution. I was wondering, do you know much about the population sizes of different branches along the tree?
Effective population sizes?
Yeah, I'm thinking one explanation for that could be different clades are responding to selection in different ways.
I mean, I'm sure it's almost
Yeah.
absolutely possible.
And so maybe if there was a way to incorporate population size, it might be interesting, but maybe it's not known.
Yeah, that's something that'll never happen.
Yeah.
It's a really good question. But
Does MCMC tree not give you effective population size on those branches the way Star Beast would?
No, it's not coalescent.
You could infer it maybe from your data because you had 6 individuals per species. So
No, that was for traits.
Oh, I'm
So
sorry.
for the molecular data, I didn't mention this. For the molecular data, I have like 3 or 4 species for which I have a bunch of samples, and then most of them, some of these specimens are only known from like 2 or 3 specimens in the collections.
Oh, okay.
You know,
There are some ways to, even with the single genome, try to, like Mike Lynch has these. But anyway, it's interesting.
Yeah, I believe you should talk about
Yeah.
that. Yeah. I think Chuck had a question back there. Did you?
Yeah, I wanted to know how does morphology or morphological characteristics map to the tree, the phylogenomic tree?
What do you mean?
Like, are they in concordance?
Oh, you mean like the osteology?
Yeah.
Not at all. I mean, it's the osteology. No one's really supported the osteology. No molecular has ever supported the osteology. It's basically useless. It's rife for homoplasia.
So diplodactyly and pygopods would not generate morphological.
Well, so from some of the older, more osteological studies, there is evidence. Like people, at least for the last like 40 years, have thought that pygopodids were somehow related to these guys, but there's no morphological data that's been published that supports the monophyly of carpodactylids and pygopodids. It's exclusion of diplodoculids. There's like one trait in the quadrate that's very similar, but other than that, it's not much. You have a question, Dave?
You have some amazing survivors on New Zealand. Tuataras, wild palmetto frogs, kiwis, things. Why didn't any lizards make it? Why did they all have to come in later?
That's a really good question. I actually don't know. I have no idea, honestly.
They're pretty good survivors. Lizards aren't good survivors, but they...
Yeah, I mean, the thing with kiwis too is they're old, but they're not that old. Like tuataras are pretty old.
Tuatara's old.
Yeah.
Wild palmetto frogs.
Yeah, they're very old. Yeah. So they're very, very old. But I feel like these guys, by the time that they're already established in Australia, New Zealand was already underwater. You know, so that might have something to do with it.
New Zealand being underwater bothers me. Wild palmettos, I have no idea.
Yeah, I mean, with New Caledonia, it's almost certainly underwater. With New Zealand, because they experienced so much orogenesis, and most of the orogenesis took place underwater.
Yeah.
During, like, I think they didn't really start having real mountain building, because New Zealand's more topographically complex in both Australia and New Caledonia. So they have peaks that are over 3,000 meters.
Yeah.
But most of those weren't built until about the Miocene, right? So it's like it's much younger mountains.
Yeah.
So it's possible that there could have been like source islands, but that's all just hand-waving hypothetical nonsense. So I don't think people really know. I don't know that much about wild palmettos dispersing there, so or being isolated by vicarians. So yeah. Tesla.
I was just wondering, you touched on this a little bit, but do you have any hypotheses about why some of these clades might have such high rates of body size change?
Well, I think at least with the island things, it has to do with ecological opportunity. I kind of try not to, like I had so much stuff here, it's really hard to focus on anything. Yeah, so I think a lot of it has to do with ecological opportunity, right? So it's, you know, your classic adaptive radiation. And I think the fact that it's only body size or one trait or the other that's being, that's varying in terms of rate at least, has something to do with constraints. So some interplay between the two. Does that make sense? No.
I was actually, looking at a bunch of these trees, I was surprised that we found no rate shifts. It's such a diverse group with so many species. I would have expected at least one.
Yeah, I would have expected that too. And I think that if you had used something, a simpler model like Medusa, which uses stepwise AIC, you would have found like a ton of rate shifts across this tree. But since you're using reversible jump Mcmc and you're able to compare across models, you know, if you don't have a rate shift across the higher proportion of the posterior, then you're not going to really detect the rate shift. And I think that the background rate for Diplodex is actually quite high. It's actually not that high. It's not like high like hummingbirds. Some groups of hummingbirds, but it's like in the middle range, right? So I think that the fact that if you have clades that have intermediate to high rates of diversification and they colonize a region which is basically open, then they can go either way. They can either increase in diversification or they can just keep doing their thing in terms of the rate. There might also be a constraint if generation time really is important in terms of the rate of diversification, which we don't know for this clade. Geckos only produce 1 to 2 eggs at a time. So, you know, so there might be some interplay with constraint there too. I don't know if that makes sense, but yeah. Any questions?
All right. Thank you, Skip.